So we've been doing this work. I've been extremely lucky. I work with some phenomenal people and some really interesting animals. So we have investigated this basically looking at decompression sickness in these animals, which previously hasn't been thought to be a problem, but there is some recent data that suggests that that may not always be the case. So this is sort of where we are. I'm a comparative physiologist, and what we're trying to do is trying to understand how physiology changes in different environments and for different species. So this is the grandfather of comparative physiology, August Krogh. So he coined the term that for every physiological problem, there's the perfect species to study. And I think if you're looking at extreme physiology, marine mammals or marine vertebrates that breath hold dive are a really good animal system because they do things at such an extreme level that a lot of things are really interesting. Acid-base balance, pressure tolerance, hypoxia, thermoregulation. So if you're interested in these things, I urge you to come and potentially work with us. So my view of physiology, I am not good at anything. I'm slightly good at a few things. I started off doing molecular biology and realized that I can't pipette, but I'm pretty good at math. So if you can't pipette, you can do molecular genetics. So I think a combination of molecular work combined with whole animal studies and then my favorite, mathematical modeling, you mold these together, you work together with different experts in different areas, in different disciplines. I think we have a much better chance of trying to understand how especially diving animals and potentially breath hold diving or scuba diving humans function underwater. As James has already gone through this, I'm just going to briefly talk about this because this is where we're going. So we're talking about decompression sickness in these animals, and what is decompression sickness? So it's basically the evolution of intravascular or intra-tissue bubbles forming due to increased levels of gases. You can either do this by going from sea level to altitude, or you can go into depth, staying at depth, and then coming back and doing this decompression. This is something that Paul Weathersby that I work with at the US Navy suggested. He said that there's basically a finite probability for decompression sickness from any decompression. So you have these tables and they're all probabilities. There's a lot of variation as has already been discussed. But there's some things that we can look at. This graph is a little bit tricky to look at, but these are species differences or basically mass differences between different species. So if you look here, you have body mass. This is a log scale. Here you have ED50. This is the saturation pressure that this species is basically getting a 50% decompression sickness risk. And these are really severe decompression sickness. They're basically, the animals are either dying, they have chokes, so they have shortness of breath, or they're paralysis. And if you see here, you have a human here, and then you have this mass-dependent risk function. So this is allometrically scaling, and something that we've recently done is looking at how does this scale with cardiac output. So most of these differences seem to be related to cardiac output. So DCS risk is related to cardiac output. What about diving mammals then? So since 1940, when this excellent paper came out by Per Scholander, we've thought that marine mammals or diving animals are basically free of decompression sickness. One, they don't carry continuous supply, it's just a finite supply. And they also have this unusual anatomy of the respiratory system. So they have a very stiff or a fairly stiff upper airway with a collapsible alveoli. So when they're going down to depth, the lungs compress, eventually collapse, and they don't exchange gas. So they then minimize gas uptake or nitrogen uptake passively. So there's this passive reduction, and you increase a pulmonary shunt, which reduces gas exchange. So this has been sort of the formal thought since 1940s until about the early 2000s, when there were reports of mass-stranded whales that had decompression sickness-like symptoms. There's been a lot of work on that since, and here you can see this is a bycaught seal with bubbles in the eye. These are turtles that we work with in Spain from fisheries bycatch, and these turtles have massive gas emboli. The definition of decompression sickness is it reverses with recompression. The people I work with had no recompression chamber, but they had an autoclave. So if you don't increase the temperature, not cooking the turtle, you can actually recompress them. So they successfully saved some of these turtles, and there's ongoing work that we've been doing with these animals showing that this is most likely a decompression sickness or gas embolic event. So what is actually going on with these animals? Why at some times do they seem to get decompression sickness, while at other times they seem to be perfectly fine? So I will show you some differences between different species, the same species but different populations. This is the Florida tourist. These are bottlenose dolphins. You have time in seconds on the x-axis. You have depth in meters on the y-axis. These are the Sarasota dolphin. This is on the Gulf of Mexico side in Florida. So they're sort of bobbing around, doing short, shallow dives, going down to this. They usually live in a fairly shallow embayment. Compared to the Bermuda dolphin, actually the Sarasota dolphin dive trace is on here. You can hardly see it. Again, time in seconds, depth in meters. These guys, the deepest dive we measured was 1,000 meters for 13 minutes. So their physiological capacity in the same species seems to be tremendous. So how do they do these things, and how can they potentially live at these extremes? So what we did was, in 2006, we created a mathematical model to look at gas uptakes and gas dynamics in these animals, to try to understand this a little bit better. It's a fairly simple marine mammal. So you have a lung, you have a circulatory system, you have a tissue. So gases are exchanged by partial pressures, and then they're transported to the tissues with a certain blood flow. At diving, the lungs are shut off, so as the diver is going down to the depth and the lungs are compressing, the pressure is increasing inside the lungs, so you get increased diffusion into the circulatory system and increased uptake. Then you reverse these when they're coming back up again. Then you add a couple more tissues, because you want to look at this effect that James was explaining at the different tissue time constants. So you want to look at muscle compared to brain compared to fat. So you're adding different compartments with different blood flows, different gas solubilities. Then we added a shunt. So basically, when the animal is diving, you can have a shunt that it's looking at how much the pressure is affecting the gas exchange across the tissues. So then we throw this into a bunch of different species. You see a Cuvier's beaked whale. These are the champions. The current dive record is 3 kilometers for over 2 hours. Here you have a sperm whale. These are the metronomes of the divers. They dive very, very consistently. Here's a blame whale's beaked whale, another beaked whale species. Here's a pilot whale. Here's a killer whale. If you look at the y-axis, which is the depth, they're fairly different. These killer whales don't really dive very deep. They dive fairly shallow and for fairly short durations of time. So you look at this, you put this into the mathematical model again, and then you predict what the tissue overpressures are when these guys come back to the surface. And if you overlay this on this figure, you would expect them to be way down here. They shouldn't have end-tidal nitrogen tensions that are really high. So this is the beaked whale for shallow dives, beaked whale for medium deep dives, beaked whale for deep dives. Here is the bottlenose dolphins, and here's an honorary marine mammal, the king penguin. They all should come back to the surface and have severe decompression sickness symptoms 50% of the time. They don't do that. So what is going on? We've been working with this for several different years, and then try to come up, we collected a bunch of different data, so we came up with a new hypothesis that basically what these animals can do, they can manage gas dynamics, they can manage the gases in and out of the lungs during the dive. So I will try to explain a little bit how we think this is going on, especially in the cetaceans. This is an older study, we reproduced this from For et al. From 1967. Here's the ventilation perfusion, so it's the ventilation in the alveoli, here's the perfusion through the alveoli. Already read? Okay. Otherwise, we're done here. You're saved, it's lunch. Or the next guy. What you have here, you have the level of shunt in the lung. Here you have no exchange of gas, so basically complete alveolar collapse. Here you have perfect matching, so the partial pressure in the alveoli is the same as in the pulmonary vein. If you do this well enough, here's nitrogen, here's oxygen, here's carbon dioxide. If you do this well enough, you can minimize nitrogen uptake or exchange, and you can still have gas exchange. So we think that this is what the animals are doing. Rather than completely shutting off the lung, they actively try to work with the ventilation perfusion. So how do they do this? Not all experiments are really good, so we wanted to understand the lungs first. So we went out in inflatable kayaks, tried to measure lung function in sperm whales. This was not a good idea. We still have not published this because we didn't get any results. So not all experiments are good. So what we did was I teamed up with an excellent researcher, Michael Moore, not a documentary filmmaker, but he's a veterinary pathologist. So we built this hyperbaric CTable chamber. Basically, it's a fiberglass tube, and you can shut it off at the ends, and then you can image through a part of the fiberglass. Then you can increase the pressure here. One of the engineering problems was this weighed about 3,000 pounds, about 1,500 kilograms, and it couldn't sit on the CT bed that well. So we had to build a frame for it. So it was slightly engineering challenge. We put dead marine mammals into this because the way that we were doing it, we couldn't put live animals. And we published this paper. So what you see here is you see the lungs at the surface, you see the lungs at 50 meters, you see the lungs at 100 meters, and you can see how the lungs are being compressed. This is in a harbor porpoise. If you look at this carefully, there's a gravity dependence on here, so the gas is going upwards. I will come back to this a little bit later. I'll come back to a comparative figure from a few different species. So instead of working with dead animals, we started working with, and instead of going out into the wild working with large, scary whales, we teamed up with aquariums and we started doing lung function testing. So what we can do is we developed this pneumotachometer, this flow meter or dolphin spirometer. These guys are trained. We could do esophageal pressures to try to look at the mechanics of the lungs. And then the animals are sitting there nicely breathing for us, so we can do some lung function testing, we can do some metabolic measurements, and we can try to understand how these animals are working. So we've done this for a bunch of different species. Just to give you an idea of the respiratory champions in the world, this is a trace from a human. This is respiratory flow on the y-axis. You have time in seconds on the x-axis. Here is a human inhaling, exhaling, and then the next breath is about here. Here's a dolphin exhaling, inhaling. This is a voluntary breath. The maximum flow rates we measured is about 150, 160 liters a second. So about 10 times higher than a human can do. About 3 or 4 times higher than a horse can do. So there's very few animals, if any, that has the respiratory capacity of these animals. So it's a pain in the behind because you need to make your own tools. There are no spirometers that can work with dolphins. So just to show you some comparisons between humans. Here are some CT scans from different species. Here's a dolphin, here's a gray seal, here's a pig. It's all at 60 psi. So if you look here, there's a gravity dependence. This animal is laying on the side because as soon as the buoyancy started changing, the animal started floating inside the chamber. So this was another challenge until we realized that you can actually attach them to the chamber with suction cups if you put suction cups on the flippers. So you attach them to the chamber and then you can have them stay still. Here is, again, gravity dependence. Gravity dependence, this is a pig, no gravity dependence. So what is going on in the lungs of these animals? So what we're suggesting is the architecture in the lungs of these animals, especially in the dolphin, is completely different. You do not have these clusters of separated alveoli. It's more like an open system with a lot of collateral ventilation. So you have ventilation between different parts of the lung. So just to show you, if you haven't heard the term collateral ventilation before, here's an alveoli, here's an alveoli, here's an alveoli. So you have these passages, these pores of cone that can open up at certain intrapulmonary pressures. Here you have them in the conducting airway, again a conducting airway, and here inside the alveoli. So what we suggested was, this is what it looks like in the cetaceans. So rather than having discrete portions of the lung that are collapsed, it's all like an open system. So we measured this in the trained dolphins using a system for humans. So what you see here is, here's a human with collateral ventilation. The blue stuff is transpulmonary pressure. Here again, transpulmonary pressure, transpulmonary pressure. This is the flow inside an obstructed alveoli. So it's basically an alveoli with a one-way valve. So the air can only go in one direction. So it's going out of the alveoli. So if there's collateral ventilation, this alveoli is still going to be ventilated from nearby alveoli, and then the air is going to flow out of it. This is the highest flow rate they can do in the human system. Here's a human without collateral ventilation. So here you see the air inside this portion of the collapsed region of the lung is decreasing. This is the dolphin. There's continuous collateral ventilation in the lung of the dolphin. So we'll come back to that in a little bit. This is the dive response in a dolphin. We have them trained, so everything we do is under voluntary control. They participate if they want to, or they can swim away. So we've made this system with suction cup based electrodes. We then can look at the dive response. So here is heart rate before the animal is diving. Here's the start of the apnea. The animal is turning over. You see this classic drop in heart rate. Then the end of the apnea, the heart rate increases. One of the things we believe that these animals can do, and there's increasing evidence of that, is they can regulate heart rate like we can regulate breathing. So they can vary heart rate and possibly stroke volume. So what we're working on is training these animals to do this under voluntary control. So the trainer gives a sign, you're going to hold your breath. We measure the breaths before going underwater or holding its breath. Here's the difference when they are in control. So this is the dive response when the animal can do whatever they want, so they can control it, and it's different at different times. I think it's pretty clear the dive response is not a reflex, because then it should be the same thing over and over and over again. We see a lot of variation in the responses, especially in marine mammals. So, summing that up, there seems to be cognitive control of heart rate, and also these animals seem to have hypoxic pulmonary vasodilatation rather than constriction, which you see in humans. So what does this mean if you have dilation of hypoxic regions? This is the difference between a terrestrial mammal and a marine mammal, in our opinion. Here's the compression of a terrestrial lung. Here's the cetacean lung. So it's this gravity dependence. You have compression on low-lying regions, and then they're sending all the blood to this region. A little bit of blood is going here, so you can still exchange oxygen and CO2, but very little or no nitrogen. So this is how they're setting this up. So when they are voluntarily and not under stress, this is how things are working. In the human, well, it's sort of going wherever it's going. So we think that this is a hypothesis that explains how animals can prevent this when they're diving naturally. And if this fails under stress, when they're exposed to sonar, when they're exposed to sudden noise, when they're exposed to man-made changes in the environment, They also send gas to the upper region, blood to the upper region, and exchange nitrogen, and then become at risk of decompression sickness. So the differences, shunt is both passive and active in marine mammals, not just passive as we've been working on for the last 70 years. And the stress alters physiology and then can explain these events of decompression sickness in different marine vertebrates. I don't have a red light, so I'm on time, right? So I work with a bunch of really, really amazing people and amazing institutions, and I thank the Society for letting me come and talk to you about this. And it's really exciting for us, like Lucy was saying yesterday. We're working with animals. You guys have really cool tools and understand physiology much better than at least I do. So it's really interesting for us to hear your opinion, and you may have some ideas on how to do some of this stuff. Would very much like to team up with you if you have interest in working with these animals. Here's some further resources. Here's my email if you have some comments. If you have, I was talking to someone yesterday about there's a mockumentary or there's a film called Blackfish. If you have some questions about that, come and talk to me about it, because I think that a lot of people are misinformed about how these animals are treated when they're in human care. This is a podcast that's doing a lot of really interesting things on marine mammals, on animals under human care. So they're doing something tomorrow talking about some of the work we've been doing. So thank you very much.